Hello, and welcome to Informed in 30, National Brain Tumor Society's education initiative committed to providing brain tumor patients and caregivers with key information about cutting-edge advancements in brain tumor research and treatment. Our topic today is understanding DNA damage response. My name is Rachel Kittleson, and I'm the Director of Community Relations at the National Brain Tumor Society and a member of our Personalized Support and Navigation team. Joining us today is Dr. Kirk, National Brain Tumor Society's Chief Scientific Officer, and Dr. Ranjit Bindra, Harvey and Kate Cushing Professor of Therapeutic Radiology and Scientific Director of the Yale Brain Tumor Center. Thank you both for joining us today.
Thanks, Rachel. I'm so pleased to be a part of this series as we launch our second year with such an important topic. It's really an honor to highlight the work we're doing with Dr. Ranjit Bindra, a physician, scientist, entrepreneur, and brain tumor leader, and the DNA Damage Response Consortium. This is really a one-of-a-kind platform for brain tumor patients and has brought together a dream team of translational, drug and treatment researchers in a world-class clinical team. Dr. Bindra?
Well, thanks so much, Kirk. I'm really glad to be here to help educate our brain tumor community on this emerging research and the potential for new treatments. This is such an exciting time to see what's available and all the new options that are on the horizon. We're really excited to tell you about it more today.
Kirk and Dr. Bindra, the National Brain Tumor Society recently announced the launch of our flagship research initiative, the DNA Damage Response Consortium, or DDRC. Our team felt it was important to use the Informed in 30 forum to help our patients and care partners understand what DNA damage response is, why this research is so important, and what the potential implications are for both adult and pediatric brain tumor patients. Dr. Bindra is the consortium's principal investigator, and Kirk providing additional scientific leadership for this initiative. I know you both have a lot to share with our community to help them better understand the importance of this work. Our hope is, as you begin your presentation, that we can give our patients and care partners a better understanding of what is DNA damage response, how do DNA damage response drugs work, what is the potential for both pediatric and adult brain tumor treatment, and what is the DNA Damage Response Consortium?
Yeah, thank you, Rachel. Again, both Ranjit and I are really excited to be able to share this information. And it's a pretty complicated area, and so really trying to understand what it is at a patient level to see why this is so important. And how we can really work to translate these drugs. So maybe, Dr. Bindra, you can start out with just a high level, what is the DNA damage response and what's the opportunity with this class of drugs?
Thanks so much, Kirk. I've been studying the DNA damage response for now, I guess, over 20 years, which is pretty wild. And I'm continually fascinated by it. And I always like to start out by telling patients that, our bodies are actually constantly under attack. It's the food that we eat, simply walking outside for sun exposure or pollution in the environment. And that means that our cells are subjected to a lot of environmental stresses as a result. At the core of all of our normal cells is the DNA, and that's the code of life. Billions of bases of DNA that code everything that is us. Interestingly, that code is constantly under attack because of the reasons that I mentioned earlier. And as such, our cells have developed a highly conserved throughout all species pathway to repair the DNA damage that actually arises under those conditions.
Oh, thank you. So that's the DNA damage response. And then when you think about what are the kind of drugs that can be utilized and leveraged, some of which have been approved for other kinds of cancers, ovarian cancer and breast cancer. Can you speak a little to that?
Yeah, wonderful question. Well, I always like to say it's probably taken, decades for us to even understand the DNA damage response, the proteins, the pathways, the interactions of those proteins at the DNA. And we still actually don't understand a lot. But we understand enough now to make drugs that target those proteins. And really in the last 20 years, we finally realized that something that's unique about tumor cells versus normal cells is, for a variety of reasons, they're unable to use specific DNA repair pathways. It turns out that those defects or mutations in those proteins, they tend to drive the tumor formation from the beginning. In the early 2000s, there were seminal papers that showed that those defects could actually be exploited, much like we think of as an Achilles heel, by using drugs that block DNA repair. Now, in the early 2000s, we've seen an explosion in the number of DNA repair inhibitors that are now FDA approved for use in patients. And one of the more common stories that you're probably aware of the use of PARP inhibitors for BRCA1 and BRCA2 mutant breast and ovarian cancer. Those FDA approvals happened about a decade ago. But a lot of these drugs, they've done miracles for many patients without brain tumors, but these drugs don't penetrate the blood-brain barrier. And so we've been unable to really access them in our clinical practice as brain tumor doctors. More recently, I would say in the last maybe 5 years, we've now understood the importance of drugs getting to their target in tumors in the brain. And we're now actually making drugs that are specifically optimized for blood-brain barrier penetration so we can start to use them for our brain tumor patients.
Thank you. So, you mentioned for ovarian and breast, the BRCA mutations. And that's an example of a precision approach and really understanding your patient population. But I'm guessing, and I know that's just the beginning of the story. And so the BRCA mutations kind of targeting those are really trying to go after a specific thing that can be Achilles heel that can be targeted. But what are the other are there other DNA damage repair mechanisms that are operant for brain tumors, for glioblastomas, for pediatric brain tumors?
Kirk, this is such a good question. This is why I'm so excited, because I'd say when I was a medical student and those stories broke in the early 2000s, everyone was saying, well, this is great, but it's only going to apply to breast and ovarian cancer. Then when I was a resident at Memorial Sloan Kettering, we started to realize, well, maybe it's going to work for prostate and pancreas cancer and other tumors. And then really over the last 5 to 10 years or so, we started to realize that there were similar mutations in brain tumors, both adult and pediatric brain tumors. And this is a story that's near and dear to my heart because our group was among a group of laboratories that reported for the first time that IDH mutations, for instance, that occur frequently in brain tumors can be targeted just like BRCA1 and BRCA2, possibly with PARP inhibitors. So now moving forward, we're starting to now look back even further and look at other gene mutations that are common in adult and pediatric brain tumors that no one really thought had DNA repair defects associated with them. But if you remember earlier, I said that those defects often are used by the cancer cells to grow and to transform quite commonly. So we've realized that many mutations in adult and pediatric brain tumors confer DNA repair defects, which means that these drugs, which are now seen as penetrant, can possibly be used against them. And as you mentioned, the key is using a precision oncology approach.
Yeah, and really, I think it gets beyond just the pathological classifications in terms of the different subtypes of brain tumors, but it really comes down to the molecular signatures, right? And so when you think about it, can you discuss some of the types of brain tumors where actually trying to develop drugs for a DNA damage repair could be a benefit, spanning from, adult indications and high-grade glioma, but also to lower-grade gliomas and then pediatrics across the board. So maybe just spend a few minutes discussing that.
Yeah, another wonderful question. Some of the simplest biomarkers that patients will see when they get that report from that first news and they open up their lab report. One of my favorite biomarkers is MGMT, or methylguanine methyltransferase, and you'll hear the promoter is unmethylated or methylated. And that simply means, is that gene expressed or not? Now, we've known about MGMT since 1980, I think we're now realizing that that in itself is a biomarker that can be used for novel combinations of DNA repair inhibitors with conventional chemotherapy and very specific types of chemotherapies, as well as radiation therapy. Other examples of drug targets are BRAF. And that's a mutation that we used to never really associate with brain tumors. It was more tumors outside of the brain, things like melanoma. We've suddenly realized that lower grade gliomas have BRAF mutations. Then there's other more less frequent rather mutations in other genes that we didn't really think were in the brain, but now that we sequence literally every brain tumor that comes to every major center, we're finding that subsets, little slices and dices of all these tumors, there often can be an actionable mutation such that the conventional standard of care, radiation, chemotherapy, may not be the only option. There could be clinical trials testing novel combinations that could give hope for those patients and their family members.
No, thank you. So when you think about the different brain tumor indications, so there's new classification of glioblastoma, lower grade astrocytomas. Their oligodendrogliomas is another space that could really benefit potentially for DNA damage repair agents. And then in the pediatric brain tumors and high-grade midline gliomas, DIPGs, those patients with histone H3 mutations. You potentially could even be moving into other pediatric brain tumors with high unmet need, like medulloblastoma. Yeah. There's just, I think that the potential here, and you discuss PARP inhibitors as one class, but maybe just give a little bit of at a high level, what are the other flavors of types of DNA damage repair inhibitor drugs that could be developed for brain tumor patients?
Yeah, so really, really great question. And start first by highlighting something excellent you said that I think would be great for families and patients to know is that really in the last 3 years, it's almost like the hierarchy of brain tumors as we know it has completely been changed to this flat platform of the molecular era. So we used to think of tumors as adult GBMs and pediatric GBMs, medulloblastoma and ependymoma. And that sort of limited us in the way that we treated those tumors because each one had a very conventional way that things were done for 50 years and we make incremental changes. And now what we're doing is looking at the molecular data behind all these tumors and realizing that a medulloblastoma may have molecular features that may make it respond just like a subset of an adult GBM, for example. So this is really exciting because it means that we can think about different DNA repair inhibitors and combinations with DNA damaging agents like chemotherapy and radiation that can cross different classical definitions of tumors. To address your direct question, not only do we have these PARP inhibitors, but now we have inhibitors that are targeting literally every single node in the step of the DNA damage response. And not to get in the weeds, but the things that we think about are ATR inhibitors, ATM inhibitors, DNA-PK, WEE1, subsets of PARP inhibitors, PARP1 selective PARP inhibitors, many, many more. Pol theta, I could probably spend the rest of the talk on that because I get so excited, but I won't. But it really is impressive, the diverse spectrum of molecules that we have now. But now, of course, the question is, how do we test these in clinical trials?
No, absolutely. And I mean, I guess that's a really good transition to why at the National Brain Tumor Society, we've assembled and built this DNA damage response consortium. And this goes back quite a ways. And I was fortunate enough to meet Ranjit, probably 7-8 years ago. And we started discussing this when I was working in an industry. And then when we started building our Defeat Brain Tumors platform a few years ago, really met up with Ranjit again and said, how can we really do this in an efficient, rational way so that, we all feel the sense of urgency. We understand particularly now because we see that some of these drugs are going to have a lot of benefit for our patients. But we want to do it in a way that really keeps that sense of urgency and systematically advances these new treatments for brain tumor patients. And so to that end, we have assembled and operationalized the DNA Damage Response Consortium at NBTS. Dr. Bindra is the scientific and clinical, really a lead of how to pull all the right expertise into one group of people. So maybe Rachel, if we could pull up the the one slide that kind of shows what this dream team of experts looks like. Aha, there we go. And so I'll turn it back over here to Ranjit in a minute to kind of talk about the capabilities, and then we can kind of go into some more detail of some of the other points that we brought up. But here's the core group of the DNA Damage Response Consortium. And again, this is a complete bench to bedside approach. It really spans all the way from young pediatric patients to adult patients. And again, I think the thing that really excites me the most is these folks have already proven themselves, and it's not about really, so this is a team science approach and working together to strategize to figure out how to do this in the most efficient way. And none of these people need to go ahead and publish at the next Nature, Cell, or Science publication. It's all about really identifying, prioritizing, and advancing the most promising treatments to patients. And you can see that there's a good diversity here geographically, and also just with what we want to accomplish. So Ranjit, why don't you just introduce some of the members here and the highlights, and then we can talk in a little bit more detail about some points here.
Yeah, thank you so much, Kirk. And, I want to start and step back, actually. One thing that I love about science is we always like to think that science just kind of moves at its own pace and things kind of fall into place. And one thing that I've learned in the last 10 years running an academic lab that is actually science is all about the people. It's the people moving the science forward. And to that point, I remember that call, Kirk, 8 years ago. I was an assistant professor, and we're trying to do a collaboration with Vertex on a DNA repair inhibitor back when there weren't a lot of inhibitors out there. And I remember meeting you at SNO that a couple years ago when you said we're going to try to get a DDR consortium together, and we'd love to have you involved. And so first, incredibly thankful for the National Brain Tumor Society for actually making this happen, because that's a group of people that are making these people, giving them the opportunity to do this. And so this consortium is quite exciting to me. Not only is it bench to bedside to biotech, it's really spans the adult and pediatric spectrum. As I mentioned earlier, we've really removed the hierarchy, and it's the molecular era. And because of that, we're going to need a team that knows how to treat adult patients, AYA patients, pediatric patients, and a team that actually knows the scientific aspects of all those tumors. And this transcends really glioma. It's all types of brain tumors, medulloblastoma and epinymoma, while the core initially is glioma. So I'd like to highlight, obviously, you've met me here, and you've met Kirk, folks like Jan Sakaria, who are the leaders of translational modeling in drug development for brain tumors. Chris Tinkle and Anang Shalat, who are really at the leading edge of pediatric brain tumor research at St. Jude. Natalie Agar up in Boston, who's developed some really innovative biomarker platforms that can be integrated seamlessly into clinical trials. Eric Solomon, a radiation oncology physician scientist at NYU, that again has developed a suite of clinical translational models that we will be utilizing. Bill Elmquist, who writes textbooks on PK/PD modeling and blood-brain barrier penetration. John DeGroot, a clinical trial powerhouse on his own at UCSF, and as well as many people know, Patrick Wen, who's really had many, many decades of experience in managing all sorts of clinical trials from the phase 1 all the way to phase 3. And Manmeet over at Miami Cancer Institute, who really understands both brain metastases and primary brain tumors, and is a clinician scientist with great energy to move the ball forward. So this group is really exciting to work with. And as Kirk mentioned earlier, the goal of this is we certainly hope to be publishing Nature and Science papers in our academic labs, but the goal of this consortium is to move the ball forward from the bench to the bedside, to de-risk assets and to work with pharma and biotech partners to get the drugs in using biomarker-directed approaches to get the best shots on goal.
Really great overview. And I think that last point that you brought out was something that I wanted to emphasize, but I had forgotten until you brought it up. It's also it's working with this group and strategizing, but also it's those biotechs, it's the pharmas, it's the biotech startups that, as we've brought this up over the last year, and when, through Ranjit's connections and through my connections and network within the industry space, now this has really resonated with these pharma companies, with these biotechs. And now they're coming to us to say, hey, we'd like to think about how we can develop our drug in brain tumors. And can we work with you to come up with a strategy? And again, this is going to be a sustainable platform. It's not just this year. It's not year one, year two. It's going to be down the road until we address every patient population within the brain tumor space with a better treatment. And so, again, I think that emphasis about the relationships with the pharma is key.
And I'll just jump in there, Kirk. Even early on in the consortium days, I think you and I, you've lived this at Vertex, and myself as a biotech entrepreneur is, not enough companies are focusing on brain tumors. Okay, in the investment community, there is a feeling that brain tumors are a difficult area to enter, and we often see it as an afterthought. And I think, particularly in the DNA repair space, where there are so many targetable mutations in brain tumors that folks on this slide have discovered, it's been exciting to sort of drum up the excitement and to help lead these folks across the finish line to bring some of their drugs into the brain tumor space.
Yeah, absolutely. And just feeding off the comments is when you think about what's been done with in high-grade gliomas for glioblastoma patients with, the clinical trial design with GBM Agile, this, global adaptive trial, that now there's actually a pathway for approvals for glioblastoma. What's lacking really is those earlier stage, the pipeline that can feed into that kind of thing. And so, as one actually really important part, the DNA Damage Response Consortium can be that pipeline for this class of drugs. Yeah. And our goal is to be not only a pipeline, so basically members within the consortium can actually run those early phase trials, show a benefit, and then it could graduate into a more registration path trial like GBM Agile. And we're also working with different groups for doing similar things with different kinds of umbrella trials or basket trials for pediatric brain tumors as well. And so I think that's really exciting. One thing that we haven't touched on yet is the potential of developing these new treatments to kind of make sure that safety is involved too, and not just giving more radiation or more chemotherapy. And this is a potential way to actually get safer drugs to patients.
Yeah. And I will second that, in that, right now what we're doing to both the adults and kids in clinic is hitting them with some very toxic therapies. As a radiation therapist, I know this, and it needs to change. Really, the hope is that this consortium will allow us to add these precision-guided DNA repair inhibitors that could reduce the doses of some of these cytotoxic conventional therapies, maybe even get rid of them for some cases. And so I think there's a lot to think about with that last comment that you mentioned.
Yeah. And then I know this is informed in 30, so we can't keep going on forever. But just before we hand it back to Rachel here to address some other questions and to summarize, one thing that that we really try to leverage a lot and really utilize within the platform is the use of patient-derived samples, so that we really we're not just relying on immortalized cell lines, but we really are trying to look at the heterogeneity um, of the cancer, but while at the same time really figuring out how to attack molecular features.
Kirk, I exactly just really agree with that comment. I think one of the things I'd love to mention is that often it's one or the other. You have labs that have PDX or patient-derived xenograft collections and want to test them. Then you got labs like my lab, which is very, very basic classical genetics, but we don't have PDX models. And so we each tend to go and live in silos and sort of go our own path. And I think what's great about this group is we've got a healthy mix of both. And the plan is to not work in silos, is to break those down, because they're not mutually exclusive. They're actually mutually informative.
Yeah, absolutely. So with that, why don't we hand it back to Rachel? Otherwise, we will go too long. And I guess that the one thing that I would also say is that we can do this on a yearly basis and give updates on how things are going, so that we can give the community an update on the DNA Damage Response Consortium. Hand it back to you, Rachel.
Thank you both so much for that outstanding presentation to help inform our patients and caregivers. Prior to the recording today, we did collect some questions from our community about this new information and how it may impact their brain tumor experience. But before I get into those, I was wondering if either one of you would mind explaining for our patients and caregivers just a little bit more about the blood-brain barrier. I know that that's a piece that sometimes our patients and caregivers could probably learn a little bit more about.
I'm so glad you asked that, because I often forget that even though it may be common knowledge to me, that it can be difficult to understand that. The best way to think about it is that the brain is a very, very sacred organ. It's sensitive to insults, to genotoxins, to the things that we ingest that go through our bloodstream. A lot of our other organs can actually handle a lot of those toxins, and in fact, our liver is designed to remove those toxins. The brain is protected because it is such an important organ. As such, it sits behind a barrier where only very specific drugs can get through the blood-brain barrier to protect the brain. And just to give folks a sense of scale and frequency, 98% of all drugs that we give in the clinic do not penetrate the brain. If you limit that to include larger molecules, it gets down to 1%. And what's very interesting is that we used to think that didn't matter, that the blood-brain barrier is disrupted because a brain tumor has already disrupted the brain, so why wouldn't that be disrupted? That, in the last 5 years, actually from consortium members on that slide, has now been disproven. We need to develop drugs that actually penetrate the blood-brain barrier, and that takes a lot of work to do that.
The only thing I would add is just to emphasize that we have some of the best expertise on the consortium that are focused in on that, and modeling that, and understanding how you get drugs across.
Yep.
Yeah, that's super helpful for the community. Again, being a community member myself, understanding that a little better is so imperative to understanding this presentation today. So I appreciate that very much. One of the questions that came to us was regarding DNA damage response. Are there clinical trials that brain tumor patients can participate in now?
Great question. And I'd say, 10 years ago, the answer may be not sure. Now there are a whole host of trials that are emerging. And not only are these trials for adults, but they're extending into adolescent, young adult population in the pediatric space. Large consortiums, and much like our DDRC consortium, are now realizing that we need to include children in these DNA repair inhibitor trials. And so there is really a large number of options for patients.
Another question, obviously our informed in 30, we would like to be even longer than 30, but if I'm a patient or care partner and I want to learn more outside of this, do you have any suggestions on where they could go to learn more about DNA damage response?
One of the greatest things I love about working with the NBTS is I can shoot an email over to Kirk and the team, and my patients can connect directly with the folks at the NBTS. because they often can help throw the football, so to speak, on who else to speak with and to get more information. Kirk, I don't know if you had any other thoughts as well.
No, I think that's I think that you're right. Absolutely. There is a lot of information, and we can point in the right direction. As the consortium evolves here in the next few months and we put up more announcements, we're going to even have a video that kind of explains the DDR and the consortium in more detail.
One of the pieces of your presentation that I just want to kind of pull on a little bit, and it is a question from our community. In terms of DNA damage response treatments, are they added to standard of care or are they given in place of it?
Great question. So typically when tumors recur or come back after standard of care treatment, most clinical trials are testing that group of patients. And in that group of patients, they're often given as a monotherapy or a single therapy or in combination with conventional therapies. As they tend to show more promise in that setting, we try to move them into the upfront newly diagnosed setting. One thing I'll also mention is we are now realizing that we may feel comfortable to directly test some of these molecules in combination with the standard of care in newly diagnosed disease. And ultimately, I'll tell you, it is my hope and dream that some of these molecules will allow significant dose reductions in some of the chemotherapies and the radiation therapy treatments that we give. And quite frankly, hopefully we'll replace them someday in some way, shape, or form.
No, I have nothing to add. I think that was perfect.
Yes, thank you. Dr. Bindra and Kirk, thank you for joining us today. We appreciate you creating this meaningful content for our community and answering their questions to help explain what DNA damage response is and how the research being conducted in the DDRC may contribute to providing patients with better outcomes.
Thank you, Rachel. I mean, again, it's great to be a part of this series and really looking forward to things coming up. And thanks to Dr. Bindra for being a part of it and also just being the leader of this DNA damage response consortium. It's an exciting time and we're really looking forward to moving things forward.
Well, thank you so much for having me here today. I just want to tell the patients and the caregivers that this is just a very exciting time. In particular, it's quite exciting to be able to walk into a clinic with a new diagnosis or a recurrent diagnosis and finally tell patients that there are new options on the horizon and they're only getting bigger in terms of the number of new options. And so it's been a pleasure serving on this session today and really look forward to continuing the dialogue in the future.
Thank you both so much. And I also want to thank our community for joining today's Informed in 30 session about the important research happening within the DNA Damage Response Consortium. We hope that you will watch our next segment coming in June, learning about biomarker testing. Thank you so much for joining us.